we have one of the best team we could possibly put together to answer every question you have about chemistry, development, toxicology, safety. Feel free, it will not happen every day. And remember, it's free. Free consultancy.
Good morning. Oh, it's a little bit low, but thank you very much for these nice and interesting presentations. I'm Siska van Doesem from ProQR Therapeutics. I remember in one of the presentations it was mentioned that oligonucleotides do not transfer the blood-brain barrier and the blood-retina barrier. How about the blood-testis barrier?
Get this thing out here. No. That neither. So we don't cross the blood-testis barrier. Quick.
I have a question for Brett. Brett, what about the data that tcDNA might cross the blood-brain barrier? Have you seen those data? Do you find that convincing?
I think it's interesting. It's intriguing. I believe that, there are portions of the brain that aren't so well protected by the blood-brain barrier, such as the hypothalamus and other regions there. And clearly we have seen single-stranded oligonucleotides, and this might be part of the explanation for tcDNA, is that we can see drug in the brain in some of these in areas of the brain that are less that are not so well protected by the blood-brain barrier. But what we get there is not effective enough. It's not sufficient enough to actually, we've been able to show a target knockdown in the brain following systemic exposure. Whether that's part of the explanation for that, or it's possible, but I don't know. But it's one possibility.
So thank you very much for all the talks. Andrew Douglas from University of Southampton. I just wanted to ask about the uptake of different cells within the brain. So you showed that, for example, in the liver, different cells are uptaking the oligo differently. And I wondered, after intrathecal injections into the CNS, is there data about different cell types taking up the oligos differently?
Well, yes, and clearly activity has been demonstrated in microglial cells, astrocytes, and neuronal cells. So really it does seem to be broadly, it's certainly broadly distributed into the brain. Distribution, obviously, in the deeper regions of the cortex are not as much as to the other regions of the cord and in the outer segments of the brain. But within those regions where we do get good distribution, clearly activity has been demonstrated in pretty much all cell types in the brain.
Thank you.
And that'll be a topic of some of the talks at this conference will demonstrate that later on this week.
Hi, James MacArthur, Sidon Development. Two questions. One, what regulates the distribution within a cell out of the organelles into the cytoplasm? Is there a good understanding of that? Also, in gene therapy, people have suggested that you get better distribution following intrathecal delivery if you put a patient or an animal on an incline table. Has that also been observed with oligos, particularly where you put them in a particularly dense solution?
So the mechanisms by which oligonucleotides are getting out of the productive compartment into the productive compartment, if you will, are just really being delineated right now, really understood. But what does seem to be clear is that the ability of an oligonucleotide to be productive requires it to escape early, fast. And in fact, we know with GalNAc, upon binding of an oligonucleotide conjugate to the acyl-glycoprotein receptor, the release is almost instantaneous upon translocation into the cytoplasm, so it entirely escapes the endoplasmic processing pathway. And we know that even without GalNAc type strategies, that it seems very early on in the endosomal process that somehow these oligonucleotides are escaping. And once they go too far, once they get into the process endosomes and obviously into lysosomes, there's no going back. They're not getting out. In the gene therapy question, for CNS.
Distribution through, from the intrathecal compartment into the brain, is there an advantage of putting the animal or a patient on an incline table?
Some of that work's been done at ISIS, also at Biogen, and it does seem to influence the pharmacokinetic distribution of oligonucleotides, and in a favorable way. So you may be able to get to deeper regions of the cortex by taking those types of approaches.
I want to add to what Brett just mentioned. With respect to the GalNAc conjugates, I think there is a natural tendency for the ligands to reach back to the receptor and then receptor being recycled back. That's one component. The second component is the lipophilicity associated with the chemical modifications. This may also contribute. But more efficient release, we see that with the lipid nanoparticles, and it is, we can follow that with very efficiently with the phosphorus NMR and so on, how this bilayer changes into hexagonal phase and how this causes the destabilization due to the charge effect as well as the membrane destabilization, which is really remarkable because when compared to the conjugate approach or the other single-strand direct approach, we see this working at 100 to 1,000-fold lower doses. That's purely due to this endosomalytic release efficiency.
Thank you.
Hi, my name is Akira Komori from OncoImmune Inc. I'm relatively new in this area, but my question is that if the modified oligo stay in the tissue for a long time, meaning not few hours or days, but several months, isn't there any effect being observed already by oligonucleotide tying up the machineries inside the cells, such as RISC complex being tied up by the oligo that introduced rather than what other oligonucleotide these machinery might supposed to work on, or dicer substrate in our large siRNA area. The normal processing involved by dicer would be prevented to occur. And I would thought that some effect of those might be appearing. And the second question is related to nucleus edge. I've been told that it's this largely localized nucleus, but protein enzyme itself are synthesized in the cytoplasm. Therefore, what is the real partition between the nucleus and the cytoplasmic area, in that perhaps antisense activities assisted by nucleus edge may be also be occurring not strictly nucleus, but also cytoplasmic domain as well?
For the last question, I can provide one part of the answer in such that in one of my first research work on antisense was inhibition of the replication of the VSV using antisense oligonucleotides. And as far as I know, the VSV cycle is completely in the cytoplasm. Nothing is in the nucleus. And still, with RNase H-mediated mechanism, we efficiently blocked the replication of the virus, meaning that there is also an RNase H activity in the cytoplasm of the cells.
With respect to the first question, at least with respect to the siRNAs, beyond maybe a week or so, it is not possible to see the compounds free, and you cannot actually detect this compound lower than detection limits. Whatever efficiency we see up to 3 months or 6 months in terms of pharmacology, that may have to do with the risk associated very small amounts. But please remember, both phosphodiester and phosphorothioates, it's a matter of kinetics. Phosphodiester's are naturally cleaved, and phosphorothioates are oxidized to phosphodiester's and then cleaved. Only sometimes I wonder is how these metabolic factors happens for the L nucleotides or PMOs and other non-cleavable linkages. I don't know how those things happen. They may get excreted as a whole molecule.
Why don't you comment on nucleoside breakdown products being substrates for polymerases and so forth?
Okay, this is something also very important question. Obviously, everybody is interested, and we evaluated that. Although Brett asked me this question, it is known that MO and LNA and CX, they are not the substrates for the polymerases, so they are not getting incorporated. But we have evaluated that very extensively with the 2' fluoros, and they are not efficient substrates either, and then we can make the triphosphates and force that compared to the natural polymerases, and they are not good substrates. So this is also, we have to address, and we answered that.
And if I could just add to Mark's comments about RNase-H, you know, clearly RNase-H is in both the nucleus and the cytoplasm. Clearly, single-stranded antisense oligos that work by RNase-H mechanism, or even the mixmers that work by splicing, are working to some extent, a large extent, in the nucleus because we can affect splicing, we can affect pre-messenger RNA, we can target introns, and so forth. To me, Mark's comments taken into account, for RNase-H single-stranded oligos, I haven't seen definitive data that they actually do work in the cytoplasm. Certainly we can target exons. We can even target exon-exon junctions, which would make you think that you can target mature RNA in the cytoplasm. But some of those oligonucleotides are such high affinity that they may be partially hybridized in the nucleus and carried out during the splicing process. I don't know. I think there's some activity in the cytoplasm, but to me, for RNase-H oligos, but I'm not convinced that it's been definitively proven.
Thank you.
Hi, my name is Petra Distra from University College London. Particular perhaps to Brett Monia, have you looked at the difference in efficiency between highly expressed genes and the target sequence of splice switching oligos against exonic sequences versus intronic sequences? Because in our experience, we've seen that with a very highly expressed genes, exonic oligos don't really work that well. And the hypothesis we have is that it gets kind of, bound to the mRNA during its travels into the nucleus.
Well, that's very interesting data, and that's data that I'm not aware of. We have done experiments to address the question of abundance on single-stranded RNase-H activity directly by using constructs to really titrate the expression level, and our conclusion was that abundance doesn't really play an effect for RNase-H, but that's probably happening mostly in the nucleus. And we've had little problems targeting, highly abundant housekeeping genes versus very rare transcripts like receptors and those sorts of things. So abundance does not seem, of the transcript, does not appear to be a big problem. I think your second part of your question was, is there preferential activity versus exons or introns? Go ahead.
Yeah, I know I'm familiar with the literature that says that exonic sequences tend to work better. We haven't necessarily seen that in our
No, I think you're
system.
absolutely right. I think that's a false belief. If people believe that exonic sequences are more sensitive than intronic sequences, that's absolutely wrong. I could tell you that I don't know this for exactly. I could tell you about our pipeline, which is a big pipeline. The majority of the antisense oligonucleotides that are in development today are targeting untranslated sequences. They're not targeting exonic sequences. There was a day, I know Art can remember, that everybody thought targeting the AUG was the best way to target antisense oligonucleotides. 3' untranslated seems to be very, very sensitive. Introns are targeting introns is very in development, those sorts of things. So I agree with you personally. I don't know.
Thank you.
Hi, Isabella Snares from Cold Spring Harbor Lab. Question for Brett. Do you see differences in the biodistribution or productive uptake between gapmers and uniformly modified antisense oligonucleotides?
For the most part, at a high level, like at a distribution level, no. You know what drives the distribution is the phosphorothioate. So my answer is based on the assumption that we're talking about phosphorothioate backbones. So if you look at a gapmer with a phosphorothioate or a uniform or a mixmer with a phosphorothioate, the distribution is essentially the same. You cannot rule out small differences in cellular distribution or those sorts of things. Now, what the other thing that the uniform oligonucleotides do is it confers additional nuclease resistance. So if you remove, it allows you to remove the phosphorothioate to some extent. by using these stable, these modifications with more greater stability. Once you start moving out the phosphorothioate, plasma protein binding changes, cellular protein interactions change, and the distribution will change. If you start removing phosphorothioates, you're going to start shifting the distribution of drug from the liver more to the kidney because plasma protein binding is reduced and those sorts of things. In the central nervous system, it's been found that there's very little CSF protein binding, and actually you don't need the phosphorothioate for distribution, and you can actually get away with phosphodiester linkages there, and you get good distribution.
Thank you.
Hi, my name is Leontien van der Bent from Radboud Institute for Molecular Life Sciences, and I have a question for Dr. Yoon about RNA sequencing as a toxicity profiling. I think it's a very exciting idea. I was just wondering, to what extent do we have guidelines for what levels of up or down regulation of immune response factors are acceptable?
So actually, I have a very few little idea about that. Since this is very exploratory field for me, so but I think we just doing like usual the drug-treated sample for this case. But I think in the future we need more the keen guideline for the oligo. But in this status, I just apply the drug-based treatment.
Thank you.
Hi, I'm Pietro Spitali from the Leiden University Medical Center. I would have two questions. One is, I saw the ICE technologies Net Generation 2.5. It's LNA with the methyl group. Did you try with the MOE instead of with the methyl group, and what changes if you tried that? And the second would be, has any of you experienced with the circular oligonucleotides?
So the constrained ethyl chemistry, it's related to LNA. It's not the same as LNA. It's a different modification of the bicyclic nucleic acid. And absolutely, we've looked at it, and others have done this. I think the microRNA companies, I know of Regulus particularly, but I'm sure the other ones too, have used different combinations of bicyclic nucleic acids with methoxy, ethoxy, MO modifications, and so forth. And depending on the situation, it can actually improve activity or improve specificity and so on. We've done it as well in our programs. In fact, we have some molecules that have reached pretty late stage drug discovery that have mixtures of MO chemistry and CET chemistry as well. And so some of the screening that Art Craig referred to in his presentation, in which you screen for tolerability and so forth, one added dimension to that we look at is mixtures of modifications. We don't just look at constrained ethyl and do lots of screens and 2' MO chemistry and do lots of screens, but we'll actually look at combinations of the chemistries and evaluate them together. As for circular DNA,
There is one circular DNA that's in the clinic. That's Molagen has a circular, it's an immune stimulatory CpG oligo with no modifications in the backbone. It's all phosphodiester. If that was linear, it would be degraded, but by circularizing it, there's no 3' end, so it is more stable than it would be otherwise. But other than that, I'm not aware of companies really focusing on circular oligos.
Hi, my name is Julie Rutte from the Leiden University Medical Center, and I have a question relating to the blood-brain barrier. Do you have any information about whether there's any uptake within the vascular wall itself, so the endothelial cells and the vascular smooth muscle cells within the brain vasculature?
You mean as part of within the structures within that constitute the blood-brain barrier?
Yes, well, I think, yeah. And also the larger arteries.
Yes, we do see distribution there. To my knowledge, we haven't definitively demonstrated target reduction in those tissues, but we do see distribution, yeah.
Do you also see it upon intrathecal administration?
Yes, we do. We see activity in those cells as well. And yeah, we do see, oligodistribution in the choroid plexus as well, with pretty good knockdown, comparable to other regions as well.
Thanks.
It's quite sensitive.
Good morning. My name is Christopher Leck from NTU Singapore, and I had a very broad question for whoever is interested in fielding it. So you guys discussed today, well, first of all, thank you guys for the very informative talks this morning. It seems like there's a very harrowing picture that you guys paint in terms of the effects of sequence, in terms of toxology or toxicity, as well as sort of the randomness by which different cell lines within organs uptake ASOs, particularly talking about antisense here. So I wanted to know, just from a broad perspective, what do you think are some of the most, or one of the most important areas where understanding can be made that will help make this design process much simpler, as well as what sort of areas do you think emerging technologies would be most useful in progressing how easy it is to, or how less difficult it is to turn ASOs into drugs?
All right. Well, I'll start it. My favorite area is endosomal escape. I mean, that's really at the heart of this, and I think everyone's touched on it, the non-functional oligo. We have all this oligo that's getting into endosomes. You can look at cells if you have a labeled oligo, and you see this ton of oligo in endosomes, and most of it never gets out. How does it get out? I think that's just a huge unanswered question, and if I had a postdoc, I'd love to put them to work on that. Do you want to?
I don't think distribution, I wouldn't call it random. We just don't understand it completely. But we know a lot. I mean, think about how much we've learned over the years on what are the most sensitive cell types. I mean, look back at some of the old literature where there's articles where the title of the article is antisense oligos don't get into cells. I mean, that's just false. We've learned so much, and we actually do know, and we have, as Mano showed in his presentation, there are so many drugs in development today, and it's not random. It's because we now beginning to understand, and I would say we understand a lot about how these drugs distribute, how they get into cells, what the most sensitive cell types are. I think Art touched on the idea of algorithms and the ability to take data. As we gather more and more data on sequencing or identifying active oligos that are most potent, oligonucleotides that are less toxic or better tolerated. I think we'll be able to develop algorithms. I think we'll be able to develop rules that will be able to select, so you don't have to do so much screening for an oligonucleotide to find the very, very best one. And I agree with Art that I think that a key area of research is being able to figure out how to shift non-productive uptake to productive uptake. And I also want to say that I think small incremental increases in productive uptake will have a major impact on activity in tissues. It doesn't take much. And if you can do that, that's where all you medicinal chemists in the audience, that's where you should focus your attention, is on that.
Okay, thank you very much.
I am Nori from Tokyo Medical Dental University. So a question for the Brett. So after intrathecal injections, you showed a very good widespread effect of the antisense oligonucleotide whole brain, including the cerebral cortex. Is it caused by just the passive diffusion of the dependent on the concentration, or some more active transfer mechanisms such as the cell-to-cell propagation or the lymphatic bulk flow in the brain?
So the distribution and activity is dependent on the natural flow of the cerebral spinal fluid. So it does track with the cerebral spinal fluid. Those cell types and regions of the brain that are most perfused with the CSF, you see an excellent correlation with activity, uptake, and distribution. As for how they get into cells, we don't know how they get into hepatocytes yet. I mean, and I suspect that many of the same mechanisms that are responsible for uptake, receptor-mediated endosomal uptake of oligonucleotides into cells in the periphery, like hepatocytes, are going to still be at work in the CNS as well. Although the cell surface proteins that are taking them into those cells will be different, but the basic mechanisms, I think, we'll be seeing. You know, we learned a lot from the whole GalNAc strategy. We really learned how, you know, for the first time, we understand how an siRNA or an antisense single-stranded oligonucleotide actually gets into cells. It taught us a great deal, not just about how to make the drugs more potent and efficacious, but also what are the mechanisms? And because we understand how receptors endocytose, like LDL receptors and those kinds of things, and how they're processed, and that taught us a great deal about how oligonucleotides must be getting into cells or some other way, or at least what we can take advantage of to improve that.
Brett, for CNS, do you want to talk about bolus versus continuous infusion? Did you mention that?
I didn't, but what we have found is that both work, and it does appear that bolus injection, we do seem to get better penetration into certain regions of the brain and better activity than
in this
continuous
case,
infusion.
the lumbar spinal cord is more actively downregulated in compared with the brain.
I'm sorry?
So I mean, are there any deliberate concentration-dependent effect by the intrathecal injections at the lumbar?
Yeah, like I said, certainly, although we can show activity in the deeper regions of the cortex, we can, but you know, you're going to get less drug there because it's deeper into the brain structures. The cord is obviously very sensitive, the lumbar regions, you know, the cervical regions, and so forth. And then as you get into the base of the brain and then into other regions of the brain that are more perfused with the CSF, you're going to get greater activity than deep down in those deeper structures.
And one more thing that you showed some, it's not big, but a slight, the downregulation effect of the intravenous injection, the antisense oligonucleotide to MALAT1. You can see some effect on the central nervous system. So are there any leak from
I think that publication really was looking at regions around the brain. It was really more of the extraction around the way they took out the brain structure as well as the cord structure. I don't believe that there's actually activity inside the central nervous system in that case. I think it's more of an extraction artifact.
Yeah, thanks.
Boeing from Moderna, and maybe my question is more specific to Mano. So for siRNA delivery, is the abundance required for the knockdown effect? And assuming the answer is yes, then have you calculated or tried to figure out how many copies of RNA is really required to achieve the long sustained knockdown? And also, at some point, to maintain the therapeutic level upon repeated long-term dosing, will you be concerned about oversaturating the risk?
Oversaturating risk. The first part of the question, namely with respect to we see maybe anywhere from 1 to 10% of the drug reaches the risk, and that's what the loaded amount is. That relates to the effects, what we talked about so far, how much is the efficiency of the release. So depending on the technology what we use, that's the efficiency, the current efficiency, state of the art. The second thing, we have not seen the oversaturation effect of the risk on any other side effects which might happen from the readouts what we can do at present.
And then you talk about lipid nanoparticles and the GalNAc. Is there any difference in terms of the affinity when they, or after the endosome escape, when the siRNA got presented to RISC complex? So between lipid nanoparticle delivered payload versus GalNAc?
Well, in terms of the GalNAc is in the sense strand, so once it's loaded, you know, the GalNAc goes its own way. Whereas in terms of the lipid nanoparticles, both strands are released, and then the common modifications what we bring, namely 5' phosphate or 5' phosphate analogs associated with the guide strand or the antisense strand, we preferentially load into that. So that's how you achieve the selectivity.
All right. Last question. So you mentioned that within lipid nanoparticles, there are about 500 to 2,000
yes.
Numbers of siRNA got loaded. Is that reflected in particle size or any other way?
Well, I think that is because of the limitation of how we make that measurement. That's more than, it's not a very precise measurement now. But we consistently see more than 95% packaging.
All right,
Yeah.
thank you.
As you know, we don't want to have any empty particles.
Hi, my name is Golnish. I'm a PhD student at Royal Holloway, University of London. I had two questions. One is quite simple. It's just about the length of the oligo. We're saying that shorter oligos are going to be more specific. This is a bit confusing for me because I was under the impression the longer you get, the more specific in terms of the shorter you are, it's more likely for that short view sequence to be repeated across the genome in various places. That's just one question. And the other one is, I don't know if you've come, of course you have, you've come across these 3D nanostructures of oligos, such as the nanopyramid of single-stranded oligonucleotides that have in prokaryotes increased intake efficiency. Have you come across that happening in eukaryotes, or is that not looked into?
So I'll be happy to answer the first one. I was careful, or I tried to be careful when I said this, to say that for a given chemistry, shorter oligos are more specific than longer oligos. If you compare a 20-mer methoxyethyl oligo to a 12-mer LNA oligo, where you've increased the affinity of the oligo to a very high level by adding the LNAs, then indeed that's not really a fair comparison. That 12-mer oligo may be able to hybridize to many more sites than the lower affinity methoxyethyl. But if you compare a, let's say, 12-mer LNA to a 20-mer LNA, the 20-mer LNA will be able to hybridize to any area in the genome that has at least 12 consecutive matches. And so there will be many more opportunities for that longer oligo to have off-target effects than the shorter oligos. We had a lot of discussion about this when the paper came out with the so-called tiny LNAs, an 8-mer LNA that was being used as a therapeutic. And many people looked at that and they said, this is crazy. This is just going to have all kinds of effects because an 8-mer, there's no specificity in the genome. There's a couple of things that make that wrong. One is that because it's a completely modified oligo, it doesn't activate RNase H. It's a steric blocker. And most of the RNA in the genome is not functional in the sense that you can bind a steric blocker to it and nothing happens. You're not affecting it. And so it's very important with sequence selection to pick areas with your oligo. You want to make sure you're not binding to microRNA seed regions. That's one of the important things with algorithms. So with steric blocking oligos, you can go very short, and you can do the RNA-seq actually with these very short oligos and show you're not having off-target effects. I don't know if anyone else wants to add on.
The way I think about specificity and length is a bell-shaped curve. I actually think if you go too long, like Art said, you're clearly going to have nonspecific effects, and it particularly is for cleavage events, for RNase H, say, for example, because as Art mentioned in his talk, if you get partial hybridization, the enzyme can be so efficient, it can cause a cleavage, and it's terminal, it's end, you're done. And if you go too short, then the possibility of the sequence within the genome of being available for binding is much higher frequency. So you can lose your specificity. And as Art mentioned, that's completely adjusted or dialed back and forth by the affinity. If it's high affinity, you can go shorter. If it's low affinity, you're going to have to go longer, and you probably have the same amount of specificity. As for three-dimensional structures of oligonucleotides in eukaryotes, I don't know anything about that. Do any of you guys? Anyone in the audience?
Good thing for you to work on.
Yeah, sorry.
I was just curious.
We're not such experts after all, Mark.
Thank you very much.
Thank you.
I have a question for everybody likes Brett, and they are asking questions about the blood-brain barrier. So I want to ask a question to Brett on this kidney cell targeting. Our compounds naturally go so much to kidney, and you pointed out nicely the localization and so on. I'm thinking why there is no oligo drug yet in kidney.
Oh, there was one.
That's, no. Yes, GLT, but I meant in completely, compared to now the liver-focused approaches. And what is it still as an issue connected with? Yeah.
Well, you're right. The first,
it's like a low-hanging fruit.
The first RNA therapeutic that came out for the kidney that I know of was SGLT2, and that actually worked in the clinic. But the program was stopped because the small molecules just took off, and they are now marketed and so forth and so on. And that's a rule for target selection: is there's no need to compete with small molecules, so don't do that because you might lose. Oligonucleotides in the kidney are generally absorbed or generally taken up by the kidney through absorption, through filtration, through the GFR, which is why you get so much drug in the S1, S2 segments of the proximal tubular epithelial cells. But there is some contribution from the blood side too that gets into the deeper regions of the kidney in the medulla area. No, I think it's just a question of bandwidth. I think the hepatocytes are a much more lower hanging fruit for targets and those sorts of things, oncology, local administration. But I do think drugs are coming for kidney. I think they're coming, especially with the new chemistries that can penetrate deeper regions of the kidney. I do think you're going to see drugs emerge that are going to be going into targeting the kidney in the development pipeline in the near future.
Hi, Jan McCaskill from the University of Edinburgh. So we've been talking a lot about functional versus non-functional delivery, and I was just wondering if any of you had opinions about how we might best measure that functional delivery, whether or not it's, Brett, you showed some amazing images of nuclear versus cytoplasmic delivery, or, for siRNAs, is it risk argo pull downs, and if you can give us some ideas about what you think the best measurements are, and whether or not we can make any technological advances there.
I'll comment and ask if anyone else wants to, but unfortunately today our definitions are functional. They're not structural, and I think that's where we need to go. I showed an electron micrograph, and we could continue to do those things, but they're obviously not high throughput. It would be very nice, and this is some of the work that we're doing, and I think others are doing too, to be able to fractionate cells into different compartments and correlate that to activity or non-activity, productive versus non-productive activity. But that work is in progress, but I think that we might be able to go in that direction in the future. Of course, you could always do visualization techniques and those sorts of things, but and that would have to be correlated with the functional activity versus non-functional, but that's all work in progress. Today, it's going to come down to fractionating and getting into the details, getting into the weeds in the cell. But today, I think their definitions really are based on function, on activity.
I completely agree with Brett, and this is a fight I've had with my drug development colleagues. Every place I've been, they always want to do biodistribution studies. We're developing new delivery systems or new chemistries, and they want to look at whether we're getting into these tissues by just counting oligo in tissue. I think that's a waste of time, because as Brett showed, oligos go everywhere. It's got to be a functional assay. I'll just add one point to what Brett did, because we looked at several different functional readouts, and we used the mallet oligo as well. That's a great oligo. Mallet is a wonderful target to knock down. If we try other targets besides mallet, we get different results, actually. And Charles Thornton has mentioned this at meetings too, in muscle, that targeting a cytoplasmic mRNA, you do not see knockdown, whereas you can easily knock down mallet or nuclear targets in muscle. And so I think that's another dimension of this that we have to think about, is the particular target that we're going after. There may be differences between cytoplasmic and nuclear targets, and it might even vary from one tissue to the next. We don't fully understand that.
Thank you.
I have just one comment and wish for that, because more recently I've been hearing from the analytical chemists, especially the mass spectroscopists, how they can do kind of MALDI imaging or even the electrospray techniques where they can do the cell fractionations and where actually they can detect the oligos. So this may be the direction the field can go and invest, so that we won't be doing the radio labeling or we won't be doing the mass balance, but just taking the cold SI or ASOs and find where it exactly is going, so that even before the functional readouts, we may be able to do some kind of bioanalytical. So this is a wish list.
Okay, very last question.
Vinashwari with Om Life Tech. Maybe I missed this. Since we talk about length of the oligos and we now know that chemistry plays a lot of role, I wonder if in your experience you have seen that shorter oligos can get easily into the cells as opposed to longer oligos, and is it a function of the chemistry as well in terms of length?
You know, the free uptake work that I showed you, the ability to just put oligos onto cells and they take it up and you can see a functional effect. We haven't seen a big correlation of length with activity. Of course, that goes to a point. If you go too low, you're just not going to get sufficient binding to the RNA to have an effect. But in cell culture, we have not seen a relationship on oligo length and the ability to take it up freely and be active. There's some belief that shorter oligonucleotides might be able to escape the endosome, early endosome, and be more functional than longer oligonucleotides. That's certainly not been proven. It's more of an idea at this place that's definitive data doesn't exist for. When you get into in vivo situations, of course, though, there's a critical length in which you no longer get distribution. If you go too distribution is driven by plasma protein binding. And I'm referring to phosphorothioate backbone oligonucleotides again. It's different for the PMOs and the non-charged oligos. I don't know how they distribute. But for the phosphorothioate-containing oligonucleotides, they're driven, the distribution is driven by plasma protein binding and prevents it from being excreted through the kidney. If you go too short, you eliminate the plasma protein binding or reduce it too much, and then you get very poor distribution. And it gets rapidly cleared through the kidney. So length for free uptake is unknown. If there's a role there might be some subtle differences for endosomal release. But once you go in vivo, you're also attached to, you have requirements for plasma protein binding that you can't lose sight of.
And a quick follow-up.
I have 2 comments. The first one is kind of an advertisement for a short talk on reversiers for single-stranded oligos, where we see GalNAc-mediated delivery, but at the same time, the shorter ones working even efficiently, so you can hear that. That's the first part. The second comment is this length dependence actually doesn't work with the siRNAs, because we need the minimum length needed for this, the 2 turns being recognized by the RISC machinery for this unwinding and so on. So it's mainly for the single strands, as you know.
Yeah, I would agree with Brett that in the usual cell uptake assays, you really don't see marked length-dependent effects with single-stranded oligos. And yet something is clearly going on, because if you transfect oligos into cells, then, and you're looking at different lengths, typically, a 20-mer, something in that length really looks best. But when you're doing free uptake studies or in vivo, that's where you see the benefits of the shorter oligos. They don't show up in transfection assays, but I don't think we fully understand yet which step it is where those short oligos are benefiting. And I'm a subscriber to the theory Brett mentioned, that it may be an endosomal escape, that might be the step where the short oligos have an advantage, but we don't know.
A quick follow-up. So would you think that this may have to do with the percentage modification on the oligo itself, as like unmodified and modified?
What modification are you talking about? We're assuming phosphorothioate backbone, but you mean like LNA or other things. It certainly could. I think everyone who's worked with these modifications has had the experience that just moving a modification 1 base over can make a big difference in the activity of an oligo.
It must be a very, very short one. Otherwise, people will start complaining that they have no time to have lunch before starting again this afternoon.
My name is Vipin. I am from RS University. I just want to know that apart from the DNA, RNA, other nucleic acids like PNA, what is the major problem for applying in therapeutics? Because there are so many papers which claims inhibition of this gene and this gene. What is the real picture actually about this?
PNAs have been shown to work as antagonisms. Mike Gate has done a lot of experiments, and PNAs have entered clinic early on with a company in Denmark. I think they have potential to work like these PMOs, but I don't think their pharmacokinetics or delivery have been optimized yet. That may be the reason. Thank you.
Okay. So, thank you again to all our speakers, and a special thank again to Korea Institute of Toxicology for sponsoring and making this workshop possible.